everybody hear me all right? That's good. So we're just going to go through two tiny examples on how you can use the function. So we're going to go quickly through the a bird example, so a ZW system, and then we'll do the possum example, which is an XY system. You can follow along on your own code. Should be fairly straightforward to run. And then the main point, so this is only going to take us, well, hopefully half an hour. And then the main point of this session is actually load up your own data and have a look, because that's the most interesting bit, is to see what you have in your own data. Because we can show you how the function goes, but you're the one who makes the question about it. So just to get started, we will just load the packages. Is that all right? Yeah. Gotta load your data. No? What's happening? Refresh. Can also just look here. So this one is the yellow-tufted honeyeater. So Deanna talked about that one, which is a bird which has a ZW system. The data set is just a simple Dart genlight object, but with no filtering done on it. So it would be, you get your raw data from Dart, you load it in, and then you run this function. So in this one, there's 609 individuals, and we have about 1,000 SNPs. We have, and then we just simply run the function. So all you need for this is in your individual metrics. So remember when they explained how Dart has different ways to access your data. So if you go into the add other, and then you look for your individual metrics, you need a column which is sex. So all lowercase sex, and the sexes are coded either with an F for female or an M for male, and anything else will be ignored. So that's basically all you need. So if you're interested for future to look at your own data, just have that column in there, sex, all lowercase, and then Dart will like when you import it into Dart, it'll automatically be in that case. So let's actually see if that one will run now. Yeah. And as we said, it's a easy function, which is GL filter sex lengths. You do need to give it your expected sex chromosome system. So if you work on species where you actually have no ID, run it twice, one which says ZW, one which says XY, and see which one gives you the best results. So as you see, for 1,000 markers, that runs really quick. And these are the results you get out of it. So the first plot you look at is your call rate plot. So since this is a ZW system, your females will have a high call rate, which means no missing data for markers on the W chromosome, so the one that is unique to the female, whereas it will always be missing in the males. So that's the first one we look at. You also notice that it's not every marker is 100% for all your females, and that's actually interesting because that's usually when there's some individuals have a mutation on the restriction enzyme cutting site, so those will actually be missing in those individuals. But overall, you'll get about that simple 0 to 1 ratio. If you have misidentified individuals in your data set, you might find them a bit higher where there's like percentage of misid'd individuals, but that's very easy to find. As long as you're not at 50-50, I don't know what's a male or a female, you should be able to get that. And this is the plot we get after it's filtered. So very nice straight line. The next thing the function does looks at heterozygosity of the individuals. Same thing. So first we're going to look at the Z-linked markers, which are heterozygous in males because they have two Z chromosomes. So some individuals will be homozygous, but if there's polymorphism on the site you're looking at, so on the SNP, then there's potential to be heterozygous, but usually only up to 50%, because that's what you expect under Hardy-Weinberg equilibrium if you're looking at individuals within one population. And the females, which only have one Z chromosome, hemizygous, will have no heterozygosity. On the other hand, and I'll try to explain again for Renee, because maybe it was a bit hard to understand gametologs. When you have some overlapping sections between your Z and your W chromosome, it will output a data object for each of your markers. So here you see we had 52 SNPs which were W-linked. So you get a data object for your W-linked SNPs. We had 165 SNPs which were Z-linked. So nice object there, and 16 gametologs, so the ones that were fixed between those. And we also found some sex-biased markers, so the ones that have a difference in call rate between males and females due to coverage. And the last one is actually the one that you're going to be interested in when you're going to do population genetics, is you get an autosomal object. So if you're just interested to do PopGen analysis, that's the object you're going to be using after you run this function. And then you go back to your normal filtering, and no issues with sex-linked markers. Everybody happy. Then the next step we're going to do, if you're interested to identify the sexes in your data, is you're going to run this function called GL-infer-sex, which takes the output of your filter sex-linked function. So it takes all the different objects we created with the identified sex-linked markers in there, and you're still going to give it which sex chromosome system you expect, so ZW in this case. And then based on that, so maybe I'll just try and run it down here. Which again, quite fast to run. And you get an overview of your identified sex for each individual. So here, each individual is a row, and then you get your identified sex based on W markers. So how many of your loci were missing and how many were called? Well, if most of them were missing, then you're likely to be a female. When you're looking at Z-linked markers, you're looking at heterozygosity, so it's going to look at your data, and you said, oh, you only had one homozygous marker, but most of them were heterozygous. All right, could be a female. And then you're looking at your gametologs, so the ones that are fixed. Same thing, we're looking at heterozygosity, so for females, you expect these to be fixed, to be homozygous. So 5 out of 5 were homozygous, 0 heterozygous, so we expect it to be a female. Between all these evidences, we assume that the agreed sex is a female. Easy. That's good if you have a lot of sex markers in your data. This is not going to cause any trouble. The issue will be if you work on a species which doesn't have a very degraded sex chromosome system, so you have a lot of homology between, so you're not going to find a lot of sex-linked markers, which is the case with some species, like some fish species. They have very small non-recombining regions. So you're going to, with some luck, you're going to find some, not many, but the cool thing is that whatever you find is going to give you a potential sex. And even if you have unknown individuals, you could at least include that in your further downstream population genetic analysis. So that was for the ZW system. We have, we'll also try this on the XY system, so the leadbeater possum. So same thing, follow along if you're going, so you can help see how the output looks in your code and how you feel about it, feel comfortable, because in a couple of minutes, it's all up to you. Go look for it at your own data. So again, when we're looking at the other individual metrics, we have this column which has sex, and that coded as F and M, and then we just run that function. But here, because we know that this species has an XY chromosome system, we put XY, and we run that. I'll just show those results down here. So this is more in my case, this is a more realistic data set to where you only find a single marker which is located on the Y chromosome. This is what I mostly find in my data sets because the sex chromosome not very differentiated from each other. But then again, it's easy to spot it, easy to identify it, easy to remove from your data. Looking at heterozygosity, so we see females on the bottom here because females have two X chromosomes. They are heterozygous between 10 to 50% expected. Males are homozygous expected. So we have some X-linked markers. Perfect. Based on that, we can, again, we get those objects for X-linked markers. So we had one SNP which was X-linked. We had 66 SNPs, Y-linked, and 66 that were X-linked. Gametolog in there as well. But again, looking at this object, the autosomal markers are generally the one that we're going to be using downstream for analysis. All right. Can I maybe ask a question for the room? Who here in the room knows the sex chromosome system of their study species? Who has no idea what they will be looking at in their data? All right. I mean, that's why we have the function. Like, it's very cool to just have a go, use the function twice, and if you find something, you're good. Because that's how I started with this type of functions. I work on sharks. Sharks, we have no idea basically what they had. So I combined data sets from 20 different species, so population genetic data sets from published or colleagues. And I basically run this function on each species. And what did I found? I found they all have XY sex chromosome system. And what I did after is like, we took the sex-linked markers and we mapped them to each other, or we mapped them to a reference genome. And we found that they're super conserved across the species, which is completely not what you would expect for a teleost fish, because bony fishes, they switch their sex system constantly. So we were quite surprised to see that, all right, these sharks, ancient lineage of vertebrates, they have XY sex chromosome system, which basically hasn't changed since their most recent ancestor. So if you have any interest in that, well, here's a good start. And collect some data from your colleagues or your old data, push it through here, have a look. So basically, yeah, it's an interesting exploratory experience. Um, just going back to the example data sets, the last bit I'm going to talk here is that, for the lead-beater possum, we ran the GL infer sex function. And you see here in the command line said there were not enough gametologs. You need at least 5. So there's no sex based on those. So when we actually look at the sex ID for that, you're gonna see a lot more of these asterisks. And that's because you only have two sources of evidence of your sex. So you have only Y-linked and X-linked markers. And basically, if they don't match up, then we are very happy about that. So then you have to make your judgment calls. And then basically from here, we have an hour left. So if you have your own data, load it up into the cloud. So get your metadata file, get your Dart CSV file, or any other sequencing file you've used, load it up in the cloud, and read your data in using the GL read Dart function. Don't do any filtering, and run this. And then there's some questions that you could try to answer, like, okay, how many sex-linked markers did I find in my data? If the answer is none, well, that's a very interesting result on its own, because what does it mean if you don't find any sex-linked markers? Does it mean you don't have enough markers, not enough coverage across your genome? Because you work on a frog and you have 30 gigabase genome. So what else could it mean? It means potentially that your sex chromosomes are not very differentiated. There are no sex chromosomes. You have epigenetic sex determination, environmental sex determination. So I think even if you don't find anything significant in the data, it still gives you information. So look at how many sex markers were present. Did you have any visual misidentified individuals in there? Because that usually happens in the field. And then you could also look at things like, did my sex markers influence my population genetic analysis? Or if you're more interested in sex bias dispersal, you could take your Y chromosome markers, your gen and object, only keep the males in your object, and start looking if there's difference between your autosomal data and your Y chromosome data, because then you're sort of looking at paternal history of the species, of the populations. So we all know that mitochondrial DNA gives you the maternal history. If you have an XY sex chromosome system, then the Y chromosome could give you information on the paternal history of the population. Same thing with X-linked markers. They're biased in the female population, so they could give you different perspective on the female history than the mitochondrial DNA, because mitochondrial is a single gene. X chromosome markers are multiple independent genes or markers. For those, I don't know if people have showed you how to upload your data to the cloud, but there's you have this file tab, and there's this thing that says upload. Then you just choose your file, so your two CSV files, and you can upload your data in there. I tried it with my data. I had I used both a 100 megabase CSV file from Dart, which took a minute, had a 400 megabase data file, which took maybe 5 minutes, so a bit of time. If you want to try it, you can also try it on your personal laptop if you're more comfortable with that. And I think
just some people would not upload the data. Yeah. So can I just check for a second?
Yeah.
Okay. Yeah. We didn't. My mistake. We didn't tell you how to install the latest versions. And the reason was because they're not ready yet. But if you want to try this function on your local computer, yeah, you can do so, but at your own risk, because the developing functions and everything we have at the moment are still buggy, yeah? I'll just show that here, and then you can say, nah, it will never do that. This beautiful Dataverse, this package has one function only. And if you run that without anything, you wait. Is it okay, or you're not out of time? Okay, sorry. So this here is on the cloud. You can see I've installed all the other packages as well, and it shows you also the version next to it, yeah? If you would run it on your local computer, you probably would only have Data base and Data data, because the other one are not installed by default. So if you want to get anything, you do this. And say you want to have Dart sex linked, yeah? And you want to have, because it's not on cron, you have to have the GitHub version, and you can specify also the branch. You want to have the dev branch. This installs the dev GitHub version on your computer, but use it at your own risk. You can always deinstall it afterwards, yeah? And that's the same for all the functions. And maybe just one more thing. If you do this, this would install the cron version of your package. This wouldn't work because it's not there, yeah? So if you say GitHub and dev, then you get dev. If you get GitHub main, you get the main, and so on. So you can install all of them. And then at the end, you can check that running the function again. Whoop. Whatever. And then it tells you what you have. Okay, makes sense?
We did forget to say that if you don't have your own data, we have uploaded our data, some example data sets, two already on the cloud file. So basically down here, we have 3 example data sets. So we have the Eastern yellow robin. So Diana talked about that. And there's some interesting, there's already some code there that you could use to run there. I've also added 2 shark data sets, so the bull shark and the blue shark, which I didn't really work out for you because it's interesting if you can just play with it. We do highly encourage to try your own data sets. And I think for this, we'll just leave it here. So you have, here's how to install it on your local computer or upload it to the POSIT cloud. And you just read your data in. I don't know if you all remember things like this, where it's tl.read Dart. And then you have your file, which is your Dart file. And your individual metadata. And basically, that's all you need after you have that GenLight object. You run our functions, and things will be revealed to you, or they won't. And we'll leave the session at that. So if you want to play with that, you have 45 minutes, and the enemy will come around if you have any questions or any issues. Same for the people online. We'll keep an eye on your questions if you have troubles. And if you just want to take an early lunch break, that's fine as well. So thank you.